Hello, my name is Nishka Jain, and on behalf of my co-authors, I'm pleased to present our project, Comparative Genomics to Discover Novel Relationships Between Sharks and Humans in the Context of Thymus Development. This work was conducted as part of the Aspiring Scholars Directed Research Program. To begin, I'll summarize our entire project. Our research is driven by the fact that sharks have a remarkable resistance to cancer, which we believe is linked to their unique thymus and enhanced genomic stability. Our motivation, therefore, was to compare key thymic genes between sharks and humans to understand the genetic basis for this advantage. To do this, we analyzed 57 specific genes across humans, dogs, and elephant sharks. We used multiple computational methods, including phylogenetics, to source data from databases like NCBI and Ensemble and map the evolutionary relationships between these genes. Our results, which I will explain soon, revealed two key patterns. First, that essential developmental genes like EGFL7 are highly conserved, and second, that critical immune genes like FOXN1 have significantly diverged, and the important signaling gene TYROBP is generally absent in sharks. Ultimately, the goal of this work is to translate these findings into innovative strategies for enhancing human immune health. So, with the complete overview in mind, let's dive deeper into the background, starting with the foundation of our work, the field of comparative genomics. This field allows us to compare the genomes of different species to understand their evolutionary relationships and the function of their genes. By looking at similarities and differences in gene sequences and gene order, we can infer functional importance. This is especially important when studying diseases like cancer, where causes and severity can differ greatly across organisms. As I mentioned, sharks are a particularly compelling model for this research. Their low rates of cancer are partly due to protective genetic mechanisms, like transposons, which can decrease the rate of point mutations. Most relevant to our work, immunogenetic studies have shown that the shark thymus, unlike a human's, does not seem to degrade or shrink over the animal's life. This is why our study focuses on understanding the nature of the shark thymus, with the ultimate goal of applying this knowledge to human health. Now, let's revisit our methodology in more detail. As summarized earlier, our process began with data acquisition, curating 57 specific genes and downloading their sequences. The second step was figure creation and analysis, where we constructed the phylogenetic trees and assessed genomic collinearity. Finally, the comparative genomic interpretation phase involved analyzing these figures to identify the key divergences and conservations that define the shark's immune profile. As we can see in Table 1, that's an overview of our generalized steps, and in Table 2, those are the 57 genes of interest that we looked at from the thymus, and we found these genes from a paper that is listed in our references. For a closer look at our results, let's look at our first major observation, and that was that some genes are highly conserved as predicted. The gene EGFL7, shown here, demonstrates this with very close relationships between the three species. This high degree of conservation is likely due to its essential role in general embryonic development, a process fundamental to all vertebrates. In strong contrast, our phylogenetic trees revealed larger differences for key immune genes. The genes for FOXN1, PSMB11, and PRSS16 all show a clear evolutionary divergence between the mammalian lineage and the shark. This repeated pattern strongly suggests that these genes have evolved potentially divergent functions related to shark immunity. However, we haven't looked into that. This was further highlighted by our analysis of the TYROBP gene. As mentioned, this important immune signaling gene is generally absent in sharks. However, there is a nuance that it is predicted in one shark species, and that underscores the importance of gene loss and how complex it is. So, what does this all mean? Our results indicate that key genes involved in T-cell development, such as EpoXN1 and PRSS16, have undergone significant evolutionary changes in sharks compared to mammals. This genetic divergence may be a primary factor in the shark's unique immune resilience. We acknowledge that the primary limitation of our study is the current availability of fully annotated shark genomes. Nonetheless, these findings highlight specific genes and pathways that should be further investigated. In conclusion, this study successfully used comparative genomics to identify key evolutionary differences in the thymus between sharks and mammals. There is still a lot more investigation to go, and our immediate future plan is to analyze the difference in mRNA levels of these divergent genes in the shark thymus compared to human and dog. Through this continued work, we ultimately hope to contribute to discovering new strategies for enhancing human immune function. I would like to extend a thank you to our research advisor, Mr. Clinton Kunha, for his invaluable guidance, and to the Aspiring Scholars Directed Research Program for giving us this opportunity. Thank you for your time, and please scan the QR code over here in the references to view our full poster. I look forward to questions.